The fifth element on the post-transcriptional regulation is on regulation of RNA stability, which I mentioned in the previous video, which is going to be a bit lengthy. So when we talk about RNA stability, so the steady state levels of your mRNA are basically determined by both their synthesis and decay rates. So decay rates would determine the time required for a new steady state to be reached after changes in transcription, and this contribute to shaping dynamic changes of mRNA levels. So, well, from this, you would know that mRNA decay rates are transcript specific and would vary over a range of up to 100-fold. So in mRNA's mammalian cells, they are typically longer lived, with half-life ranging from less than 20 minutes to several days. So overall, eukaryotic mRNAs are protected from exonuclease degradation by the 5' methylated guanosine cap. Okay, so remember your cap and also your tail. So these are the ones that protect them, okay, from degradation. So degradation, somehow, regardless, okay, would start with the removal of one or both of the protective structures, either the cap or the tail or both, followed by digestion through the action of 5' to 3' of 3' to 5' exonucleases. So your enzymatic activities is associated with cytoplasmic mRNA decays, and they are performed by a small number of protein complexes. So deadenylation is carried out by 3 different deadenylases: CCR4, NOT, PAN2/PAN3, and PRN. So decapping by DCP1, DCP2 heterodimers, okay, 5' to 3' degradation by the XRN1 exonuclease, and 3' to 5' degradation by the cytoplasmic exosome. So each and every one of them have specific protein that have their function, okay, to degrade. Now, the different steps of mRNA decay, okay, which involve deadenylation, decapping, and exonuclease degradation, takes place with transcript-specific rates. And the stability of a specific transcript is at least, of course, partly controlled by cis-acting sequences. So, but this is frequently, but not always, located in 3' untranslated region or UTR. So these sequences motifs usually act by binding to specific sequence-specific RNA binding protein, your RBPs, and these RBPs, okay, would in turn modulate the interaction of your mRNA with elements of the core degradation machinery. This is an example. Many mammalians, the mRNAs actually contain sequences, okay, called AREs, okay, or AU-rich elements in their 3' UTR. So this actually makes them unstable. So exosome, okay, is required, okay, for rapid degradation of these mRNAs, but it cannot recognize them on its own when mRNA contains ARE. So ARE binding proteins associated with these sequence elements, okay, will basically recruit component of the core degradation machinery, including the capping enzyme, the CCR4 NOT, the deadenylase complex, and the exosomes plus XRN1. So basically, apart from that, other ARE binding proteins are could stabilize their targets. Well, exact molecular mechanisms are still unclear, still being studied. However, okay, this effect could be mediated through protection from your miRNA mediated degradation, microRNA. So RBPs function, okay, as mRNA turnover and translation regulatory, okay, whereby or TTR, okay, so your RBPs are basically emerge as pivotal post-transcriptional regulators of gene expression. So we know that, okay, RBPs are major regulators of mRNA stability and translation, apart from being for the initiation of transcription. So, for example, RBPs are present in your mRNA, such as the 5' cap structure, okay, at your 7-methylguanosine, remember from the previous topic, and the 3' poly-A tail, plus a group of mammalian RBPs are associated with specific mRNA sequences, okay, at the cis segment, which are frequently present in the 5' and 3' UTR. So it could also affect changes in mRNA stability and translation rates. So traditionally, mRNA stability and translation would be the determinants, okay, of AU-rich element and are often included the pentamer triple UA. But growing numbers of mRNA stability and translation determinants has illustrated, okay, over the past decade that many examples of regulatory sequences have been described, okay, whereby they actually lack, okay, AUUA pentamers and do not exhibit a particular AU richness. So the term RBP is deeply ingrained in the literature, although it is deeply ingrained. So RBPs usually are implicated in stabilizing or destabilizing of RNA, and of course in promoting or suppressing the translation. So the group of TTR RBPs comprises of RBPs implicated primarily in the mRNA decay. So these are tris tetraproline (TTP), AU binding factor AUF1, also termed heterogeneous nuclear riboprotein HNRNP, D. Okay, the K homology splicing regulatory protein KSRP, and butyrate response factor 1 BRF1. So other TTR RBPs such as Hu proteins, QR, HuB, HuC, and HuD, and nuclear factor 90 and F90 can enhance the stability of target mRNAs and in some cases influence their translation. So before you ask, I would say this, because previous students would ask, do we need to remember all this? By right, you do. Okay, it's just that probably usually, well, now that we have an online assessment, okay, online test or online exam, probably okay, we could ask this, okay, but if we were to ask you to remember it, memorize it, usually it would be impossible because there's too much, okay, in RNA stability to remember because it involves a lot of protein to stabilize, okay, the mRNA. So another group of TTR RBPs have been shown to inhibit protein translation, and this would include T-cell restricted intracellular antigen 1, TIA1, and the TIA1 related protein, TIAR. So in some instances, these multifunction TTR RBPs usually have been associated in pre-mRNA splicing and combined DNA, and of course, okay, influence transcription. So almost all of the aforementioned TTR and RBPs are known to be shuttling proteins, and as a shuttling proteins, their function are closely linked to their transit between the nucleus and the cytoplasm. So for RNA stability, it could be achieved by methylation, only these three processes, turnover, and degradation. So degradation, of course, as commonly used by small RNAs. So we'll go for each and every one of them. Methylation protects RNAs from uridylation and truncation that leads to the degradation, as you can see here. Okay. So in plants, in flies, okay, and also in animals. So this is basically done differently, methylation, as you can see. So basically it is methylated, okay, but methylation occurs basically differently and using different proteins, other proteins involved as well. Okay. So second, turnover would reduce accumulation of naturally methylated RNAs, and 3' nucleotide addition would affect the stability of RNAs. So your poly A tails, okay, would sometimes lead to further RNA degradation for certain RNA isomers only through polynylation, though polynylation actually do have a protective role against degradation, but sometimes they probably have no effect on stability of RNAs. So protein factors can also stabilize or destabilize RNA-induced silencing complexes risks to influence the accessibility of nucleases to RNAs. But most RNAs would lose methylation activity, okay, which basically causing them to proceed to turnover. And when it goes to turnover, eventually, okay, it will cause exonucleotic degradation. So this is basically all three of them at one go: decapping, okay, deannulation, and endonucleotic pathway cutting off. Okay, so decap, okay, would basically go into exonucleotic decay. The same goes with deannulation, okay, would basically cause, okay, or deannulation sometimes, okay, it will be decap and goes to exonucleotic pathway, okay, or sometimes, okay, during, okay, the poly A shortening, okay, it goes to exonucleotic decay, okay, which is done by exosome, and this is actually the simplest pathway, endonucleotic, because it will just cleave, decay, that's it. So RNA degradation is, well, somehow a prevalent activity. So there are three major classes of intracellular RNA degrading enzymes, either ribonucleases, okay, so RNases. So it is they are endonucleases that cut RNA internally, that's like endo, okay, and 2 exo cut externally, 5' exonuclease hydrolyze RNA from the 5' end, and 3' exonuclease which degrade RNA from the 3' end. Now why can't you use one exonuclease, okay, for both 5' and 3'? Try and find it out. So endo And 3' exonucleases have been characterized in all domains of life in every organisms. But 5' exonucleases were believed to be absent from bacteria. But in 2014, there are reports that it may also occur in bacteria. It's just that no follow-up after the one paper in 2014. So, several different classes of RNA degradations are, the first one is processing, whereby we know that essentially all RNA species are synthesized as larger precursors. Hence, they must undergo 3' and in many cases 5' processing by nuclease activities. So many are excised piece of fragments, which must be degraded just like introns excised from your mRNA precursors. So such maturation pathways are integrated to the processing of all classes of RNA, regardless of mRNA, tRNA, RNA, and removal of the discarded material probably would terminate total RNA degradation. Secondly, mRNAs and non-protein coding RNAs and cRNAs, the regulated turnover of mRNA is a key factor in the control of gene expression. And of course, this is an apparently universal feature of mRNA metabolism, which is actually not really well understood. So large numbers of unstable ncRNAs, non-protein coding mRNAs, basically undergo rapid and continuous degradation. The third is quality control. Surveillance pathways usually appear to be active on all classes of eukaryotic RNA, which constantly identify and degrade defective RNAs and RNA protein complexes. This is of course to protect and to make sure that your gene could function and produce well-functioning protein. So most RNA surveillance occurs only on defective RNAs or ribonucleoprotein particles, which are presumably quite rare. Usually it's on RNAs. So mRNAs with premature translation termination codon (PTCs) generated by your alternative splicing or by programmed genome rearrangement in some specific cell types usually are target for nonsense-mediated decay and MD pathway. So quality control, when you talk about quality control, it's the same as when you work in a factory or anything that is under quality control. It's the same mechanism. So this is the process of processing and also the degradation. So each and every one of them, capping, elongation, splicing, and termination, being described to you. So I'm not going to go into details on this. You can read about it in your notes. So anything that you don't understand, we could probably discuss on it later on. So the same goes during export and also translation. So we know that when it crosses the membrane, so this is mostly it would involve crossing the membrane as well, but not so much as in the export and also translation. Now, what would be the role of stability changes in gene expression? Is it very important? Because there are so many things to know. There are so many proteins involved. So changes in stability of any particular RNA would result in two common features, as you can see in your slide and in this video. Changes in mRNA stability often accompanied by parallel alterations in transcription rate of corresponding gene. And when mRNA stability is regulated, it usually involves a protein, which is of course the end product, with rapid and transient change in the synthesis. So alterations in RNA stability could be used in transcriptional control if transcription is shut off in response to withdrawal of a particular signal. So there you have it, the lengthy part of RNA stability. So the simplest that I could summarize, because actually RNA stability is very complex. So I just summarize each of the steps, and basically on the methylation, turnover, and also degradation or decay, plus in all the steps or during the transcription process up until the translation. Alright,